Welcome Dipanjan Roy from IISER. Dipanjan, you can share your screen and present.
Yeah, okay.
Thank you.
So is the screen visible?
Yes, it is visible in full screen. Can you just
Yeah,
hide
thank you.
the notification?
Okay.
Yeah,
Yeah.
thank you.
So good afternoon, everyone. This is Dipanjan, and the topic on which I'm going to deliver a talk today is anisotropic cytoskeleton structure regulates cytoplasmic physiology along apical basal plane. So the whole topic can be divided into two parts. In the first part, I will talk about the cytoskeletal filaments such as actin and vimentin and their differential distribution in different planes of a cell. And in the second part, I will talk about the cellular rheological properties in different planes. And at last, I will try to correlate uh, these two data. So let us begin at first with by discussing about the cytoskeleton. So the cytoskeleton is a network of intracellular fibers that gives a cell its shape, and it is mainly comprised of three components. They are actin, or more commonly known as microfilaments, microtubules, and intermediate filaments. So among these three types of filaments, I have mainly worked with actin and vimentin. Vimentin is a kind of a intermediate filament. So let's first discuss about actin. So this is a cartoon of a cell, and if we see a cell from the side like this one, then we can divide the cell into three sections along the z-axis, and these three sections are the basal plane, mid plane, and apical plane. So the distribution of actin and the structure kind of actin filaments are different in these three planes. So I have also put three representative images of actin. So in the basal plane, we can see that there are a more number of stress fibers and long actin filaments. But in the mid plane, the actin filaments are not that longer, and the stress fibers have disappeared from there. But and in the apical plane, there are no filaments at all. Only some short strands of clusters of actins are present there. Similarly, in case of vimentin, we can see that in the basal plane, vimentin has the most dense network, and when we go from basal plane towards the apical plane, the density of the vimentin network decreases gradually. So this is by looking by bare eyes. So how do we quantify the complexity of these cytoskeletal networks? For quantifying this, I have you measured the fractal dimension values of the cytoskeletal networks in each planes, and if we see these graphs, that we can see that both in case of actin and vimentin, the fractal dimension values they decrease gradually when we go from basal plane towards the apical plane. Here, in case of actin, we can see that the highest FD value is almost 1.42, whereas when you go from basal plane towards the apical plane, it decreases to 1.25. Similar thing happens in case of vimentin also. So this calculation were done in control cells. So next, I wanted to investigate what happens when we disrupt the cytoskeletal filaments. Here I disrupted the actin filament with the help of cytokalasin D, which is a very well-known and potent disruptor of actin filament. So by treating with cytokalasin D, we can see that the stress fibers have disappeared completely, and instead of any kind of actin filaments, we can see some very short clusters. So this is visible. This is very well visible from the images, and I also wanted to investigate what happens when actin is disrupted. What is the of actin disruption on vimentin network? So here from this image of vimentin network, we can see that in control group of cells, the vimentin is mostly nucleated towards the nucleus, and it is not very much well spread towards the periphery of the cell. But after cytokalasin D treatment, the vimentin network it also spreads towards the periphery of the cell. So once again, to quantify these two phenomenon, I have once again calculated their FD values. And in case of actin, we can see that the cytokalasin D treated cells they had the lesser FD values in each and every plane. But in case of vimentin, we can see an opposite trend. Here, by treatment with cytokalasin D, the FD values of vimentin has increased in midplane, and it has and it has kept increasing up to the basal plane. So I had hypothesized that maybe the disruption of actin filament causes the overexpression or upregulation of vimentin network inside the cell. So to confirm this, I had performed an Western blot experiment also. And from this Western blot experiment also, it can be seen that in control group of cells, the expression level of vimentin is lesser than the cytokalasin D-treated group of cells. So from this, it can be hypothesized that maybe the actin disruption causes overexpression of vimentin, which helps to compensate for the loss of actin, and it helps the cell to maintain its structural rigidity. So that's where the rheology part comes. So the rheology of the cell can be measured using different techniques, but I have used particle tracking microrheology. So it is a passive method of measuring cell rheology, and one can use a atomic force microscopy also, because that is a more direct method. But one drawback of atomic force microscopy is that we cannot measure the rheology in different planes of a cell. So that's why I have gone with the particle tracking microrheology. So what is this? Basically, in this method, some fluorescent submicroscopic beads, which are 200 nanometers in diameter, they are ballistically injected inside a cell, and then the movement of these beads are tracked for a specific time period. So I have tracked the movement of the beads for 20 seconds, and then I have plotted the trajectory of the beads, and using a code, we can convert this trajectory of the bead into an MSD curve. So MSD curve means mean square displacement curve, and just by looking at the MSD curve, one can get an idea about the viscoelastic properties of a cell cytoplasm. So here at so let us begin with at first by with the control cells. So I have measured the rheology in control cells both in the basal plane and in the mid plane, and after analyzing the results, it can be observed that the mid plane, it has a higher MSD value compared to the basal plane, and higher MSD value means that this plane is softer. So we can say that the mid plane has less elasticity than the basal plane, or or in simple words, we can say that the mid plane is softer than the basal plane in control group of cells. So next, I wanted to investigate what happens when we treat these cells with cytokalasin D and disrupt the actin filament. So here, in case of cytokalasin D treatment, an opposite phenomenon is observed. Unlike the control group of cells, in case of cytokalasin D treated cells, the mid plane becomes stiffer than the basal plane, and it has a higher elastic value. The mid plane has an elasticity value of 69 pascals, whereas the basal plane has an elasticity of 29 pascals. So why did this phenomenon occur? So for this, we had hypothesized that maybe vimentin is responsible here, and it is playing a key role here, because we have seen previously that disruption of actin filament causes the upregulation of vimentin in the mid plane. So maybe this upregulation of vimentin in the mid plane is helping the cell to become helping the mid plane of the cell to become stiffer than the basal plane. So to confirm the role of vimentin in cell rheology, I had knocked down the vimentin from the cell line using siRNA-induced gene silencing method, and then these vimentin knockdown cells were also treated with cytokalasin D. So now this group of cells, they lacked vimentin, and their actin were also disrupted completely. So when we measured the rheology in this group of cells, it was observed that this group of cells, I mean the cells which were vimentin knocked out and cyto D treated, they had the softest basal plane and mid plane. Both in the case of basal plane and mid plane, the elasticity value were the least for them. So by doing this experiment, by doing this measurement, it can be confirmed that maybe the that vimentin definitely plays a key role in maintaining the cellular rheology, and the knockdown of vimentin affects the cell rheology. So these were the work which were done up till now, and the inferences that can be drawn from these are the disruption of actin filaments results in increased expression of vimentin, as I have told earlier. And both actin and vimentin have significant roles in maintaining cellular rheological properties. And loss of cytokalasin D treated cells become stiffer at mid plane because the loss of actin filament results in increased vimentin expression. So this was a kind of half of the work, and there are some future plans of work. Till now, we have only probed the role of intermediate filaments and actin in maintaining the cellular rheology, but we have not looked what is the role of microtubules. So that is a plan to do in future. And another thing is we want to observe the rheological properties of a cell in 3D microenvironment, because in cell culture dishes, the in microenvironment is not 3D, it's kind of 2D. So what happens in 3D microenvironment, that is another thing to look out for. And the last thing that we'd like to observe is the role of plectin in maintaining the cellular rheology. So plectin is a kind of a protein which interlinks the all of the cytoskeletal components such as vimentin, intermediate filaments, actin, and microtubules. So would like to see what happens when we knock down plectin and what is the effect on effect of this in cellular rheology. So finally, I would like to thank my PI and all of my lab mates and CSR and ICS for providing financial support. Thank you.
Thank you, Dipanjan. I think there is a question from Dr. Bidisha Shina. What is the level of heterogeneity in these measurements?
The level of heterogeneity is, if we see the Edor bars, so it can be seen that almost in each and every cell showed the same phenomenon. And although there was slight, very less variation, but the trend in almost each and every cell were same. So very less heterogeneity was observed.
Okay. Okay. So I think there is no more questions.